life, and both the origin of life and the synthesis of life. Before I do that, we have in town the Santa Fe Institute Complex Systems Summer School. Shout out, yeah, where are you? This is the next generation about to take over the world. And so you ought to know we can take them all out. So the question is, what is life? Most of our interest in space, if we were honest, is finding another form of life. And the question is, how rare is it likely to be? Do we have some set of principles that would tell us that we should expect to find it? Have we built it? Have we built software that's already alive? Some of us believe we have, others would say no. It has to be based on chemistry and capable of open-ended evolution. And so this panel is going to address this issue of, if you can't find it, maybe we can make it, and maybe it will replace us, which by the tone of the previous panels might be a good idea, actually. So let me just introduce the panelists quickly. So over here we have Kate Adamana. She is an assistant professor of genetics and cell biology at University of Minnesota. I'm not going to give you their backgrounds, it doesn't matter, but just to say that she works on the experimental synthesis of life in the laboratory. Sitting next to her is the great David Baum. David, that means tree in German, and David wrote a book on trees, which is sort of weird. But David is a professor of botany at the University of Wisconsin-Madison, and is trying to build, I guess, a little mineral surface on which life will just spontaneously emerge. He will talk about that. At the end, we have the two moderators. We need two, because this is a very difficult and delicate topic, and someone might get very upset, so it's policed by two individuals. Chris Kempe is at the end there, who is one of the faculty at the Santa Fe Institute, who works on scaling laws, universal scaling laws, to include the fundamental constraints in the evolution of life on Earth and elsewhere. And next to me, Alexandra Witz, who is a co-moderator, who is a contributing correspondent for Nature and Science magazine, and with her husband Jeff, who is down there, I believe, in support, is the author of Island on Fire, about the 18th century eruption of the Icelandic volcano Laika. So join me in welcoming our panel.
Well, thank you very much for coming out. Thanks, David, for the nice introduction. He was talking about life as software. I sort of think about life when I'm writing about biology, which I don't do often, but I try and use the recipe analogies. I usually talk about cooking things from scratch. So what are those most primordial building blocks of life? What do we need to put together in a test tube to make life? Is green chili the basis of all life? Is that the most fundamental thing in our recipe? It just might be. So we're going to talk today about not just planets as sort of inert balls of rock and gas, but planets as living things, right? The skin of life on every single planet that we know of. We hope, we'll see. The question though is why? If we want to think about why life has arisen and what it might be like, and what it might be like when we go and look for it and try to find it, what if we could do it ourselves in our own test tubes, in our own laboratories, with scientists like these sitting on the stage? Why do we want to do that? What is the fundamental thing that we could do in our labs that we can't do, that life itself can't do elsewhere? So really, I want to see if I could start with Chris to talk about why we might want to build life from scratch. Earth evolution has done amazing things over 4 billion years. Why do we want to build it from scratch in the lab?
Yeah, I think that's a very interesting question. So for me, I'd say my overall interest in life is to try and understand what the phenomena of life is as a general process. So we have general theories of physics and chemistry to a general theory of life. And you know, such a theory should be able to tell us how life started, where else it will be, how easy it is to form, how many different ways it can form, what's the maximum amount of diversity, all these sorts of questions. And so I'm very excited about the possibility for us to build life from scratch, so to speak, or to build it in the lab. Possibilities for us to test new principles, to take a general theory and use it in a new context, one that we can control, one that didn't come from this spontaneous evolving process on our own planet. And I think that comparative technique has been immensely successful for sciences like chemistry and physics. And even in biology, much of what we know about life comes from this comparative perspective. So I think the possibility of designing or creating a new form of life opens up the space of comparison, and that may really help us to further test or understand the principles underlying life in general. And I want to mention a little bit something that David alluded to, which is that in some ways we have created forms of life already, and we've learned a lot from that. So in silico evolution, so that's evolution inside a computer, simulated life, artificial life as it's sometimes called, has been an ongoing field, and much of what we know about evolutionary theory and population genetics comes from these simulations that we've built inside computers. And so I think we already have built strange forms of life, and we've learned a lot about what evolution is in its most general terms. And so my hope is that building life in the lab will help us to understand more of the sort of generalities that we expect to see in the living process throughout the universe.
Thanks for that, Chris. I'd like to ask our other panelists to talk a little bit about why you do what you do. Why do you think we want to build life from scratch? What inspires you to get into the lab and try to dream up what evolution has already done here on Earth? Let's start with Kate.
I think all of current biological sciences that's been done so far are kind of boring because all of that research has been done on a sample size that equals 1, because we known life form right now, our own terrestrial modern life that we study. And I, as a chemist, I'm used to thinking I need bigger sample sizes to be able to generalize. And you cannot generalize anything about life by studying single life forms. So that's why I'm trying to build artificial designable living systems, because I think once we actually have different ways to do life, we'll be able to generalize. There are things about our own life that are unique versus there are things that are general principles of this phenomenon known as life.
And David, you spent a lot of time looking at the evolution of life on Earth as we have it now and how we've gotten to the diversity that we see today. What inspires you to look beyond that?
I think there's several other reasons that I already mentioned that really are very motivating. You know, when we look at life as we know it, it's very complicated, and we always wonder as evolutionary biologists, how does complexity arise? And just to understand that process in the ideal way would be to learn how to recreate that process, to actually find ways that the building blocks of life can organize themselves without our intervention into things that have the fundamental properties of life, that they can propagate themselves in space, they can grow, reproduce, and that they can evolve adaptively. And I can't really think of a much better way or any better way to understand that than to figure out how to do it ourselves.
Right. Let's talk a bit about how rare life is then. What are the fundamental ingredients that we need to have a living creature, and how common are those ingredients throughout the universe? Let's start with you.
Well, so, I mean, that's almost it's almost an unanswerable question until we understand what it takes to make life. I have a biased position. I actually am of the view that life is easy, and life of the most basic kind is easy. I think that because all the life we know is super complicated, that you have cells that have these boundaries around them that are tremendously complicated in regulating what comes in, what goes out. They have this genetic system, which is also very complicated. We have this idea that it must have taken some phenomenally rare and special circumstance for this thing to arise. But if we kind of get down to the basics, a cell is just a set of chemicals that somehow work together to make more of the same set of chemicals in their neighborhood. And with that much sort of softer understanding of what life is, I have believed that life of that basic kind could emerge really quite easily, without cells, without genetics. And I tend to think in terms of things like just sort of slimes of organic molecules on mineral surfaces that can spread and grow and evolve in that setting. And so if you take that view, then life of that really basic kind, I think, is probably abundant in the solar system and in the universe.
So you're a life optimist then, it sounds like.
I'm a life optimist if you take this very sort of low-level understanding of life. I don't know how many places have genetic life, let alone intelligent life.
Okay, so we might not have interplanetary festivals everywhere, but we might have microbial mats everywhere.
Yeah, there's a good chance that we have, at least cells everywhere.
Okay. What do you think, Kate?
We know that we know that universe is lousy with organics. Everywhere we look, we find spectroscopic evidence that there is organic molecules. And I'm there with David. If there are organic molecules and liquid water, there's going to be life. And it's going to be primitive and simple, but it's going to be ticking. And the question is how unique our complex life is, and that's something we have no answer to. I actually think the complex life, like cellular and multicellular life, is rather unique. Hard to get there. It's easy to get started. It's easy to get a simple protocell going. But if you want to evolve something more complex, that's rather hard, which is, I think, this is the only interplanetary festival, at least for a while, in this part of the universe. But there probably are at least microbial life forms elsewhere, and I would have a hard time believing there aren't, given all the evidence we have about the physical chemical conditions elsewhere.
Okay, so another vote for simple life almost everywhere. You work at a complexity institute, Chris. What do you think?
Yeah, so to use your term of life optimist or pessimist, I think I'm a life pessimist 4 days a week and a life optimist 3 days a week.
Okay.
And the reason is, on one hand, I would say, we have one lineage of life that we see. So if we look at modern life, it all shares the same genetics. It shares the same basic machinery, especially the really complicated things that exist inside a cell, like these amazing structures, ribosomes. Those all seem to have one evolutionary trajectory, and so it points to everything being part of one lineage, which is interesting. And so, on one hand, I would say, the fact that we don't see other lineages could be taken either pessimistically or optimistically. The pessimist side would say, well, we see lots of exclusion in ecology. We understand how some life forms exclude others, and extinction is a very common story in the history of life and in the modern world. And so one lineage doesn't tell me life, it just says that lineage was more successful over lots of others. On the other hand, we also know lots of cases where you can get coexistence. And so the fact that we don't see a second lineage coexisting that's very different makes me maybe more pessimistic that it's actually hard to have multiple origins, or it's hard to have an origin in general. And so I don't think we have a full theory of what that overall ecological evolutionary process looks like for, say, multiple lineages evolving at the same time and competing against one another. And so I'm not really quite sure until we have that whole picture whether I'm really optimistic or pessimistic about how easy it is to form life.
So this is fascinating.
Yeah.
So we've established that life, in a very simple form, might be common throughout the universe. It may not be. Complex life might be much more difficult to evolve. But what fascinates me is that scientists today, including some of you sitting on this stage, are trying to reinvent the wheel, right? Start with a whole new set of ingredients and build life from nothing. So I'd like to talk a little bit in some slightly gory detail, but please remember, not all of us remember what ribosomes necessarily are. A little bit of gory detail in the types of things that you're doing in your laboratory. What are you putting together to create life? How do you know when you've gotten to life? And how successful or how far do we to build life from scratch? I'd like to talk with Kate. Can you talk a little bit about what you're doing in your lab at this very moment? You have a Build a Cell initiative.
Yes, so we take non-living components. Most of the time they're very complex non-living components. They're proteins, nucleic acids, and lipids that make the membrane of most modern cells. And they're very much dead by the time we get them. So we work with kind of purified compounds. We're putting them together in a way that hopefully will give rise to some emergent properties. And I'm not saying life. Obviously, I would love it to suddenly learn how to be alive, but our systems are not yet. We're putting together those lipid membranes that are very much like cell membranes. And then we're putting proteins and DNA and RNA inside, and it starts doing its thing. It starts doing what it's supposed to. For example, those ribosomes, which is the machinery that makes proteins in cells. If you put it inside a lipid membrane of that synthetic cell, it will start doing what it's paid to do. It will start making proteins. If you put genes inside those synthetic cells, they will be expressed. So you can have whole genomes. Those genomes can be expressed, proteins can be made, metabolism can be jump-started. And so we are reconstituting some processes of life. Not all of them. For example, we don't have self-replication yet, but we do have very crude evolution. We do have very crude homeostasis. I don't think those systems are alive yet, but some people argue with me and say they totally are. It's, I guess, personal, depends on your personal definition of life. But we're doing it to have an experimental model of life. Because if we look at life cell, we cannot fully describe it. We don't know every single molecule that is in life cell. But in our experimental systems, we do know every single molecule because we make them from scratch. So we know everything we put in there, and then when we see some activity, we can at least try to derive that activity from the building blocks, and that helps us understand better what does it take to do life on the molecular level.
And can I just follow up? You said it depends on how you define life. How do you define life?
I try not to, because every definition I can come up with, someone will prove wrong. So I think I'll know it's alive when I see it. Obviously, there's the classical NASA definition of life, which is a self-replicating chemical system that undergoes Darwinian evolution. And it's a great definition, but it's not an experimental definition. When you look at something in front of you, for example, I look at David, he does not undergo Darwinian evolution as we speak. He had in the past having his kids, but right now he doesn't. But that doesn't mean he's not alive. So that shows you shortcomings of that definition, and in the same way, there are shortcomings of every other definition of life you can come up with. So that's to your question, and the short answer is I don't have a definition of life.
Okay. I'd like to go to David because we don't know if he's alive yet or not. So it seems like
I'll pretend.
Good transition to see if a non-living being can come up with an answer. Tell us about what you're doing in your laboratory to basically create life from scratch. Tell us what you're working on.
So, I mean, in some sense, what I'm doing is complementary to what Kate's doing. So, if you think about what Kate's doing, she's taking components from life and trying to put them together and engineer a way to put them together so that they can reignite as they live. One of those candles that you blow out and they come back on again, in a way. And we're taking a sort of a very different approach. We're just asking the question, what if you just take the chemical building blocks of life? We know a lot about, we have a good understanding of how the primordial Earth would have been full of organic molecules, and ask the question, can they spontaneously organize into systems that self-propagate and have the potential to evolve effectively? And so we're really, the motivation for that is, if we can get these systems emerging using the chemistry of our planet, we can actually do the same experiments using the chemistry of other worlds. And so you may wonder, like, okay, how would we do this? The reason I'm doing, I should tell you, I'm trained as a botanist. I spent my PhD was climbing trees in Africa, doing pollination biology. So I wouldn't be doing this if the experiments weren't ridiculously simple. And so the experiments that we're doing approach this in a slightly backward way. So we reason, if you make a soup, and on the surfaces of mineral grains in that primordial earth, organic systems organized and began self-propagating and evolving, how would we know it? Well, one thing we could do is we could actually tell them to tell us that they're doing that, and not worry about their chemistry. So we're not looking for genetic signatures. We're looking for a behavior at a larger scale. So I actually brought some props. Not very exciting props, little vials, okay? So we make these little vials that are like a little microcosm of the primordial Earth. They have an atmosphere like the old Earth, primordial soup in a seawater mix, and then they have lots of little tiny microscopic grains. And what we do is we incubate the soup together for a period of time, and then we transfer a small piece of that into another vial that contains fresh ingredients. And we keep doing that over time. And the reasoning is that if at any time a system emerged or nucleated on a mineral surface like a slime and could grow and could move from grain to grain, then it would propagate itself. And we would know it because if it propagated itself fast enough, when we see the next generation, it will look different because something will have grown. And so we do this sequentially, and we've been finding that in a particular combination of a primordial soup and this mineral, which is iron pyrite, fool's gold, very appropriately named, we are getting these signatures that tell us that some self-propagating system has emerged. What it is, we don't yet know, but we're doing all the experiments to try and figure that out. But it's very exciting to see some indications that, contrary to the idea that these simple systems are difficult, that these simple systems seem to just emerge in a very simple experiment that you could do in a high school lab.
So that's pretty incredible that you can just hold. I'm actually thinking about that rock right
the rock.
there as kind of a basis of background, so I like rocks, but to go from something like this and to add some ingredients and then get some type of, whether it's life or however you want to define it. Chris, I wonder if I could ask you to pick that up and talk a little bit about what this might mean for origin of life. I mean, if we can go into a laboratory and put these ingredients together and start to get some type of behavior, however you want to call it, what does that tell us about the origin of life, either here on Earth or elsewhere in the cosmos? Is it easy to do?
Yeah, I mean, I think if this works, then it says a lot about how easy something might be to do, right? How easy it might be to form life. I mean, I think in general, a big problem for origins of life research has been the space of chemical possibilities is very complicated. There's very even when we write down very simple equations of just a few chemicals, we can often get very rich dynamics in that. And so the upside of that is it's easy to form patterns, and that's nice, and patterns may be the way that you then start to get structure and evolution and replication and all these sorts of things. And so that's the hopeful side of what David's trying to do, that if you can get that going and understand some of how that happens, or narrow the parameter space as we understand it, to force a certain sort of evolution to happen, then that gives us a lot of hope. I think in general, the challenge is the parameter space is big. How many distinct regions of that parameter space give rise to something like life, and how do we search that space? And so selection and evolution is often a really nice way to search parameter space, and so I have a lot of hope for the types of things that David's doing.
Right. If I can just stay on that origin of life topic for a second with you, Chris. I mean, there were sort of those classic chemical experiments in the 50s, Miller-Urey, attempting to of life, which had their flaws and turned out not to be an accurate way to describe the rise of life on Earth. Do you think we're closer today, and actually maybe we'll open this to the whole panel, in terms of what we can do in the laboratory? Do we have better techniques to replicate what might be the origin of life? Are we better now with our tools and our ingredients than we were decades ago? Do we have the technology to figure this out?
Yeah, so I'll defer on the chemical and biological technology to David, but I would say one thing that has changed from the 50s is our understanding of the dynamics. So what we've understood, in the mathematics of dynamical systems, has allowed us to go from something like Miller-Urey, where you're just putting a bunch of stuff together and trying to add a lot of energy and have a rich mixture and hope something comes out of that. Since then, we've understood a lot about dynamical systems and how to search in complicated sets of dynamics for pattern formation, for replicating processes. And so I think actually a lot of what's happened on the theoretical or the mathematical side helps us to better design things a priori, so that we can hope to get structure formation or replication. And so then combining that with the chemical perspective, hopefully will lead to something fruitful. But in terms of the chemical side, I would definitely defer to Kate and David.
Yeah, Kate, can you talk a little bit about what you're able to do in the lab? Maybe not at a real granular level, but how close are we to trying to build life from scratch, to succeed in what you're trying to do? Will you do it next year, 5 years, 10 years?
So I'm up for tenure in 3 years, so that's my deadline. But seriously, I think we're very close and very far. That depends on how generously do you want to define life. If you want to build a system that sort of resembles a living system, so it has a compartment, it looks like a cell, it might be able to divide, we're not very far from that. But if you want to build an actual complex ecosystem, if you want to build cells that can interact with each other, that can learn, that can change their environment, for that we're still quite ways away. And another question is how honest are we at doing it? Because, for example, what David's doing right now is very honest prebiotic chemistry. He takes elements that could exist on Earth before life started, And he uses only that to put together his systems. What I'm doing is totally cheating, because I'm taking elements that need biology to be made. We're taking very complex proteins, very complex lipids that need a biosphere to be made. And what we're learning is how to build life from elements that are already primed for life, that are already complex enough. And what David's learning is how to build life from the very first principal building blocks. But the living systems that either of us can build will be very different. Mine will be sort of like a bacteria if we succeed. His will be who knows what, a completely new lineage.
A slime or something, yeah, like I say, yeah. Yeah, I mean, I think there's a couple of important points there. I mean, first of all, what Chris said. So one thing I didn't mention, so this project I'm doing actually emerged out of an initiative from the National Science Foundation and NASA, and it involved the three of us actually, among other people, getting stuck together in a not very good hotel in Maryland for a week to brainstorm how to do these experiments. And so we collaborate on this research that I'm the PI on, but they're both co-PIs on. And so I just think it's important to be aware of that. And the way we convinced NASA to fund this with a not insignificant amount of money was to say, chemical parameter space is big. In order to be able to search that, we just need protocols that are easy to do and so we proposed just developing protocols. We never claimed, I don't think, that we would find self-propagating, evolvable chemical systems. The idea was just to figure out the ways to do this in practice. And it was almost like, one of those weird, I mean, to me, maybe it's too good to be true, but it felt like one of the very first experiments we did, which we intentionally modeled after what's known about the prebiotic Earth, was this one that started showing this, what looked like an ecology. And just your original question, it's interesting because I'm an evolutionary biologist and I've run a plant genetics labs for years. So I think a lot about DNA, RNA, development, but when it comes to life, I actually think that we would have been almost better off if DNA hadn't been discovered. Because what happened is that once Miller Urey did these wonderful experiments about how the early Earth would have been, what it would have been composed of, the natural follow-on would be experiments like we're doing, which is to ask, can they organize themselves into self-propagating systems? But just then, in time, DNA was discovered, and the entirety of the biology community was like, oh, okay, shiny thing over there. We're going to focus the next, 50 years of research on genetics, to the point where people consider DNA the stuff of life, but you can't take DNA and make life. A lot of life is in the protoplasm that is basically being self-propagating itself and growing and dividing for billions of years into all these organisms, and that is not all DNA. So I actually think we almost might have been better if we'd started in the 60s.
I think you were the first biologist I've ever talked to who said he wasn't so sure that we wanted DNA to be discovered.
Well, not there,
just
wait a little bit, yeah.
Yeah, just a bit, a bit.
Yeah, and I actually, just to jump on that, I completely agree with David on the DNA side. And part of the perspective that we've lost in biology with the discovery of DNA and all of these detailed molecular mechanisms is the search for sort of the large regularities of life, right? And so thinking about what the regularities for life in general are is something that has been left behind, because we can probe the very detailed function of a single gene and we can sequence lots of organisms. But that left behind a lot of really interesting research about what cells of really different size do, and what are the physics affecting cells, and what's the bulk physiology and function of an organism. And a lot of that, sort of got dropped in the 60s and early 70s, and we're just coming back to it now in really fruitful ways. And some of those things teach us more about what life is doing and what we should expect in general. And so I think that there are very exciting avenues that genetics derailed for a while. Genetics also gave us a lot of power in other dimensions. And certainly made evolution very explicit, and a lot of what we've learned about evolutionary history has come from the genetics, but the sort of what I would call macroscopic biology has been left sort of uncovered for a while because of genetics.
So maybe building life is doing a recipe with different ingredients in different patterns maybe we focused too much on one area and not looked enough at all the possible combinations of how we can put together all the different ingredients to create an end product at different places and different scenarios is kind of what it sounds like. Yeah.
Yeah, I mean, I can imagine, I mean, there's no reason to assume that life has to have a digital genetic system at all. I don't think it's required. I'm not even convinced it's required to have a cell membrane. So, although I do think cell membranes would arise. But yeah, so I think that we have to remember that we have just this one kind of life we know, and if we really want to see how many of its features are necessary for life and how many are just accidents that happened during our history, we have to do it again, I think.
Okay. I'd like to wrap up by asking just sort of a broader question about, why we're doing this in the first place and what sorts of practical applications there might be for trying to develop life from scratch. So there are sort of all the philosophical things about, should we build life in the test tube? What would that look like? Are we going to build creatures that will take over the universe, which it sounds like we're not going to do anytime soon, based on your lab work. But in the interim, what are the types of things, the types of practical applications to help us here on Earth? Can we build better drugs? Can we make ourselves live longer? Are there implications for what you're doing that might actually benefit us here in 5 to 10 years?
So we're actually working along those practical lines. I love the big picture questions, but the practical applications are paying the bills. So we're using those synthetic engineered cells, drug synthesis and therapeutics platform. So you can think of those synthetic cells as kind of non-replicating, non-threatening, but intelligent little nanorobots. And we're looking at possible applications in targeting cancers, especially solid tumors. We're also looking at point of care and remote medicine, not only in remote locations on Earth, but also for long space exploration. As of last year, we demonstrated that you can actually launch synthetic cells into space and the rocket doesn't fall apart. We've done some first kind of Sunbeam rocket tests and it works. So we're kind of trying to develop it as a platform for synthesizing biologicals and nutrients during long-term space missions, as well as point-of-care medicine on Earth in places where it's hard to set up even a little portable lab. We're also looking at using that platform for drug discovery, because synthetic cells are much simpler than natural cells, but you can recreate some of the natural cell processes. So you can take a synthetic cell and build, let's say, an oncogene pathway in it, and then try to study what small molecules tickle that entire pathway without all the endogenous stuff that usually goes on in a cell.
So you could build drug factories in space.
That's kind of the overarching goal. One of them. Also biomaterials. Since synthetic cells are already dead, if something's dead, you can't make it any more dead. So you can make materials that normally would be very toxic to live cells. You can make those materials in synthetic cells. One day when we run out of oil, we're going to run out of petrochemicals. So we need to find a way to make all of those very toxic but necessary chemicals. And that would be one of the things where that platform would be useful too.
Synthesizing fossil fuels instead of mining them from the ground.
Yeah.
Yeah. David, what about your lab? What are you going to...
Well, so, I mean, obviously for us, the big motivation is to understand how these things happen and to understand, from that, where else, on what other worlds would we find similar processes. But in the process, if we do succeed in finding heterogeneous chemical systems, so chemical systems where no one chemical replicates itself, but a whole set have this property where each one helps the other ones be made. So these, if we find these systems that can do that and can evolve adaptively, then in principle we could, find ways to, instead of designing chemicals for particular targets, for particular purposes, trying to breed chemical ecosystems to solve particular problems. You want to get rid of a pollutant, so instead of trying to design one magic molecule that will do that, you would select upon a whole community of interacting molecules for that set that not only can self-propagate, but can also evolve to get good at doing that task. So in principle, you can imagine a future where we don't have plant breeding and animal breeding, we have plant, animal, and chemical breeding.
Wow, that would be amazing.
That would be kind of cool.
Yeah, you could have a farm for the chemicals.
Mm-hmm.
Chris, I wonder if I could ask you to kind of round up for all of us. What do you, in terms of where do you think this field is going in the next 5 to 10 years? What do you expect we might see coming out of these laboratories, and what might we be learning about that, about our own life on Earth?
Yeah, so that's a great question. It's a big question. So I'd say, I think there's real hope for building better theories of life, and I think as David and Kate both alluded to, there's real hope for potentially seeing different types of evidence for life in the universe, ranging from looking at the atmospheres of exoplanets to sending probes in our own solar system. And I think a lot of that hinges around the sort of need to have, as I said at the beginning, new types of theory and new types of experiments, new types of playgrounds to really understand what life is. And even from this perspective of applications, I think a lot of the main challenges we face have a huge biological component. So whether that's antibiotic resistance in bacteria as they evolve and respond to our antibiotics, whether that's the connection between the human microbiome and health, whether that's climate change, all of these have huge biological interfaces. And so I think understanding more about biology, having better theories for biology at all scales helps us to address those challenges, and has the potential for real discovery in terms of understanding the full history of life from the very simple abiotic origin to the current complexity we see, and then also where else and how likely we are to see something somewhere else in an astrobiological context.
Right.
So I think lots of interesting things happening in the next 50 years around the living process.
Right, so stay tuned.
Yeah.
Great. Okay, well, thanks to the panel. We're out of time, but we do have 10 minutes for questions. So I believe, do we have